Hello everyone, so today we're going to talk about the resting membrane potential and the action membrane potential. But before we talk about that, I would like to show you the parts of the neurons so you could just follow up later when we say the names of the parts of the neurons. First thing that we're going to talk about is this part here. This is called the cell body or the soma, and it is the life support center, and it's responsible for the gradient potential. And inside it here it has the green part here. It's the nucleus, and the nucleus houses the genetic information. We're not going to really talk about it a lot in this video, but the one that we're going to talk about like a lot is this part here, which is called dendrites. The dendrites receive messages. It's called the receptive region because it receives a lot of messages from like the neighboring neurons and stuff. And the most important part here is this part. This is the axon hillock. It's the most important part because this is where the action potential generates in every nerve cell. It's also called the trigger zone. And right under it here, this long part here, this is the axon. And the axon, so the action potential goes from the axon hillock to the axon terminals. This is called axon terminals. And here there are different colored parts. This part here is called nodes of Ranvier's, and this part here is called Schwann cells. The Schwann cells, the yellow part here, are formed of myelin sheath, and this insulates the potential, the action potential, and allows it to have really fast connections. The myelin sheath, the nodes of Ranvier here are not covered by myelin sheath. It looks like Tic Tacs, and it's like a space between Schwann cells. The last part of the neuron here is the axon terminal, and it's called the secretory region because it releases neurotransmitters to the neighboring neurons.
Hey guys, now that we all know what a neuron is, we're going to talk a little bit about resting membrane potential. When a neuron is at rest, it's negatively charged on the inside, as you can see here I drew a negative charge, and positive charge on the outside. So it is important that we know this because when we talk about action potential or nerve impulses, then we're talking about a temporary switch of electrical charges where the inside becomes positive and the outside becomes negative. So these two would end up somehow switching. So we need to understand when the neuron is at rest, the cell has what is called a differential charge in the membrane potential. Membrane potential has the ability to do work. So since there's a separation of charge, we can create a nerve impulse. Resting potential is pretty funny to call it resting potential since it does use a lot of energy and the neuron works hard just to maintain that resting membrane potential of positive on the outside and negative on the inside. So I want to be more clear that resting membrane potential has a lot of sodium on the outside and a lot of potassium on the inside. So as we know, the sodium-potassium pump is the main reason resting membrane is even possible. The pump gets its name because it literally pumps and helps push sodium out of the cell and against the concentration gradient and pulls the potassium back in. To do all of this, the pump requires lots of energy. So it requires a lot of ATP to do such. ATP has 3 phosphates, and when it loses one, it then becomes ATP. The pump does all of this to keep the neuron at resting state, which is at negative 70 millivolts. The sodium-potassium pump is found by the axon hillock. So ATP attaches to the pump, releasing the phosphate. ATP then turns into ADP. Once the phosphate is released, the pump then changes its shape of the channel, allowing the sodium ions to leave the cell body, and the potassium then goes into the pump. The pump then lets go of its phosphate, which lets the pump reset to its normal structure, releasing releasing potassium back into the cell body. By doing that, it keeps the electrical charges where it needs to be for the neuron to be at resting point, which is negative 70 millivolts. So now I'm going to show you a video how the sodium potassium pump works. So as you see here, the pump is at its original structure. 3 ions of sodium come in, ATP then attaches, releasing 1 phosphate. As it changes structures, the 3 ions exit the potassium pump back out of the cell. And now it is prone to have 2 potassium ions leak back in to pump. The phosphate is then released, which allows the structure to go back to its original form, and then potassium is released back into the cell body. And by all of this happening, it allows the cell to be at negative 70 millivolts, which is at resting state.
Okay, now that we've learned what resting membrane potential is, let's learn about action potential. So action potential occurs within a neuron, but where? Where in the neuron? It happens here in the axon hillock. So it starts here and moves all the way down the axon into the terminal branches, and the axon terminals it's secreted. So action potential is the message that occurs in an axon, and it's transmitted to another neuron. When negative 55 is attained, it has reached depolarization, which has means everything has switched. So sodium has rushed in. If we look here, this is where depolarization would occur. Then as you go down here would be repolarization, because it's just trying to get back to negative 70, which is resting membrane potential. And then when it goes down here is hyperpolarization, and it's just reaching itself back up. It went way down low, and it's just trying to get back down, back up to negative 70 millivolts. Okay, so action potential is achieved by beginning with negative 55 at the threshold. From negative 55 millivolts at depolarization, it reaches up to 30 millivolts, and at 30 millivolts it hits action potential. Then from there it goes down to repolarization. At repolarization, this is when the sodium channels are closed and the potassium channels are leaky. They're kind of sort of open, and then it goes down to hyperpolarization. Hyperpolarization is when cell membrane potential is around negative 90 millivolts. It wants to go back to negative 70 millivolts, which is resting membrane potential. So that is when the sodium potassium pump starts working.
Okay, so in order to summarize everything that we said earlier, resting potential is all about being positive from outside and negative from inside, because we have more sodium outside and more potassium inside. Once you get a strong stimulus enough to reach the threshold, which is negative 55, depolarization starts to happen, and that makes the sodium channels opens, making the sodium rushing in. And that makes the cell membrane, inside the cell, it's positive and outside is negative. And once it reaches the peak, which is around 30 millivolts, the sodium channel starts to close, and then the potassium starts to leak out. At the end of the action potential, it reaches hyperpolarization, where we have the sodium, most of the sodium is trapped in the cell and most of the potassium is trapped outside the cell. This is where the cell wants to go to negative 70 again instead of negative 90, and this is where the sodium potassium pump starts to work, pumping 3 sodium ions out and 2 potassium ions in.